Hello students, today we will be talking about enzymes. We will be discussing classification part in detail. Now first of all, what are enzymes? The term enzyme was basically given by Robert Cuny. This particular term, that is enzyme, literally means in yeast. Summer and Meyerbeck, they were two scientists. They defined enzymes as simple or combined proteins acting as specific catalysts. Now, some of the salient features of these enzymes include they are proteins in nature, they have a proteinaceous nature. Secondly, they are biological catalysts, they are soluble, they are colloidal molecules produced by living cells. They differ from catalysts in the fact that they do not change the chemical equilibrium point, but they only speed up the rate of reaction. That is very important with respect to enzymes. Now we'd like to classify enzymes, which is our main topic. Now classification has to be on certain basis. It can be done on the basis of substrate acted upon by the enzymes. In case of enzymology, there are two things. Enzyme needs a substrate. Substrate means on the that is the chemical substance upon which it acts. The substance upon which enzyme acts, that is known as substrate. So it was Duclax who named enzymes by adding the suffix "ase" in the name of substrate catalyzed. For example, carbohydrases. The suffix has "ase", ase. Carbohydrases indicates that it is an enzyme which acts on carbohydrates. So carbohydrates is the substrate here. Similarly, proteinases. The suffix "ase" indicates that it is an enzyme. Proteinases means those enzymes which act on proteins. Similarly, lipases means which act on lipids. Nucleases are those enzymes which act on nucleic acids. Maltase is the enzyme, it acts on substrate maltose. Similarly, sucrase acts on sucrose. Urase means it acts on urea is the substrate. Tyrosinase, it means it acts on tyrosine. Similarly, lecithinase, it will act on lecithin. So as you can see from the figure also, enzyme needs a substrate. Enzyme has to bind with the substrate. Enzyme binds with the substrate to form enzyme substrate complex. That enzyme substrate complex dissociates into enzyme and free enzyme and the products. So this is one particular classification on the basis of substrate being acted upon by the enzyme. Other classification would be on the type of reaction catalyzed by the enzyme. For example, hydrolases. Hydrolases clearly indicates that class of enzyme which catalyze a particular chemical reaction, that is hydrolysis. Similarly, isomerases means that class of enzyme which involve which reaction? Isomerization reactions. Similarly, oxidoreductases means those enzymes they would catalyze oxidation reduction reaction, means type of reaction. These are specific to that particular reaction. Dehydrogenases means they would catalyze dehydrogenation type of reactions. So they are specific to dehydrogenation reactions. Similarly, transaminases means they would catalyze transamination reactions. Transketolases, they will involve transketolation reaction. Phosphorylations mean those very enzymes they would catalyze phosphorylation type of reaction. So they phosphorylases are very much specific to phosphorylation reactions. Similarly, we have another classification on the basis of chemical composition of enzymes. For example, we have enzyme with protein molecule only, pepsin, trypsin, urease, papain, amylase. Enzymes which are proteins, They also have a cation. For example, carbonic anhydrase is an enzyme. It has a zinc cation along with it. Arginase is an enzyme. It has manganese cation with it. Now, enzyme molecule has a protein part as well as a non-protein component that is called prosthetic group. So here we have different classes of enzymes. For example, iron porphyrin enzymes means it has a metallic group, that is non-protein part, that is iron, and other one is the porphyrin protein part. Similarly, you have flavoprotein enzymes, diphosphothymine enzymes, pyruvate mutase, etc. So this is a classification based on chemical composition of the enzyme. The next we are concerned with one more important classification, that is on the basis of substance being hydrolyzed and the group involved. So this is based on chemical reaction, that is hydrolysis. We have a particular substrate that is getting hydrolyzed and the type of group being involved. For example, we have carbohydrate hydrolyzing enzymes that includes glycosidases, beta glucuronidase. Secondly, we have protein hydrolyzing enzymes, we have endopeptidases, we have exopeptidases. Lipid hydrolyzing enzymes that includes lipases, esterases and listhinases. Next we have oxidoreductases, hydrases, mutases, oxidases, dehydrogenases and peroxidation. Lastly, we have some miscellaneous class, that is catalase, carboxylase, carbonic anhydrase, transpeptidase. So this classification is based on the substance getting hydrolyzed and the particular group involved. Now we have one more classification that is very important, which is actually practically followed, that is called IUB system of classification, International Union of Biochemistry. This classification, no doubt it is complex, it is descriptive, but it involves a number of enzymes. Enzymes have been grouped into 6 major classes. Now each class has 4 to 13 subclasses. Next point, each enzyme, it has two parts. One is name of the substrate. Other part, second part ends in suffix "ase", means type of reaction catalyze. Now each enzyme has been assigned a systemic code number which is known as enzyme commission number, that is EC number. Now EC number for each enzyme has series of 4 numbers at 4 places. Now number 1, first number, indicates the major class to which the enzyme belongs. Then we have two median numbers. Second one indicates subclass and the third one indicates subclass of the enzymes. And the last number or last digit indicates serial number of enzyme with subclass. Now an example we have here, EC means enzyme commission number 2.7.1.1. Now the first digit 2 indicates it belongs to major class, that is transferase. That is the major class here. Means it is transferring something. Some group is being transferred by this enzyme. Now the second digit, that is 7, that indicates subclass. Now it is transferring what? It is transferring, say, phosphate group. Then you have the third digit 1 indicates subclass, means an alcohol group is being taken as a phosphate acceptor. If it is transferring some group, there has to be another group to accept that. Here it is alcoholic group that would act as acceptor for the phosphate molecule. And the last digit, final digit, indicates the name of the complete enzyme, say hexokinase or ATP dehexose 6-phosphotransferase, means the substrate is hexose dehexose, phosphate group is being transferred, phospho, and transferase is the major class of the enzyme to which it belongs. Now this enzyme clearly indicates that it catalyzes the transfer of phosphate group ATP from ATP. ATP means adenosine triphosphate. It is a high energy phosphate. It has 3 phosphate group. This is being transferred to hydroxyl group on the carbon atom C6 of glucose. In first reaction of glycolysis, we get glucose 6 phosphate. ATP is very much involved in that. Phosphate group is being transferred and we get first product glucose 6 phosphate. This is being indicated by final digit. Thank you. Now, next important class is first class of this International Union of Biochemistry, oxidoreductases. Now, this oxidoreductases is the first class. Second one you can see transferases. Third one is hydrolases. Fourth one is lyases. Fifth one is isomerases. And last one is ligases. Now the first class is oxidoreductases, means this class of enzyme catalyzes oxidation reduction reactions of CHOH, C double bond O, CHCH, CHNH2, C double bond NH groups. Now enzymes acting on CHOH group of electron donor. Now the example is 1.1.1.1. This enzyme is basically alcohol NAD oxidoreductase. Alcohol is the substrate, NAD is coenzyme, nicotinamide adenine dinucleotide. Reaction involves is oxidation reduction catalyzed by this oxidoreductase enzyme. Other example is enzyme acting on CH group of electron donor. Again you have this electron, this EC number of the enzyme 1.3.2.2 acyl coenzyme A, cytochrome C oxidoreductase. You can see this reaction. Acyl coenzyme A is acting on cytochrome oxidase, giving you 2,3-dehydroxyacyl coenzyme A plus reduced cytochrome C. So this cytochrome gets reduced to reduce cytochrome C. Now the first digit means it belongs to which major class of enzyme? That is oxidoreductase. Now we have the next class, transferase. Transferase is that major class catalyzes the transfer of a group other than hydrogen. Enzymes catalyze transfer of one carbon group. For example, aldehyde group, ketonic group, alkyl group, phosphorus group, sulfur containing groups. For example, we have EC number 2.3 acyl transferase. 2 indicates it belongs to which class? Transferase is the main enzyme. So what it is doing? It is transferring. What group is trying? Acyl group. So you have the EC number 2.3.1.6 means acetyl coenzyme A, choline O acetyl transferase. Acetyl coenzyme A is reacting with choline. You get coenzyme A and O acetyl choline, means this particular choline is being transferred to acetyl choline. It is a transferase. Now class number third is hydrolase, means those enzymes which catalyze the hydrolysis of their substrates by adding water across the bond they split. So substrates are esters are catalyzed by hydrolysis, ethers are catalyzed by hydrolysis, peptide bonds are catalyzed by hydrolysis, acid anhydrides, carbon-carbon single bond compounds, halides, phosphorus nitrogen bond is being broken down by hydrolysis. Now the EC number would be 3.1.1.1. 3 is the major class to which it belongs, that is hydrolyzed. 1 is the subclass and 1 is another subclass, and 3 indicates the final name of this particular enzyme. Next we have lyases, that is enzymes acting on carbon-carbon single bond, carbon-carbon oxygen bond, carbon-nitrogen, carbon-sulfur bond. Carbon-carbon lyases, for example, Enzyme number is 4.1.2.7. 4 is the major class to it belongs. 1 is the subclass. 2 is subclass. Last digit is the final name of the enzyme. For example, we have ketose 1 phosphate. Enzyme is dihydroxyacetone phosphate involving an aldehyde. Next example is carbon oxygenase 4.2.1.2 L-malate hydrolase. Another example is fumarase. L-malate is being catalyzed by enzyme fumarase. Next example would be isomerases. That is that class of enzyme uh, which catalyze racemization reactions, which catalyze isomerization reactions. That includes racemases, epimerases. For example, EC number is 5.1.1.1. Example would be alanine racemase. L-alanine is being converted to D-alanine. This is an isomer. Cis-trans isomerases. Next class will be ligases, enzyme catalyzing reactions forming carbon-oxygen, carbon-sulfur, carbon-nitrogen, carbon-carbon bonds. Here the EC number is 6.2.1.1 acetate, that is coenzyme A ligase AMP. AMP means adenosine monophosphate. These are also referred to as synthetases. Now, in short, we have more than 3000 enzymes known. Oxidoreductases, hydrolases, transferases predominate. Conclusion is official names are lengthy, trivial names are generally used after identification. This is the four types of classification of enzymes. This very classification is accepted IUB system, International Union of Biochemistry. Thank you.